Hey everyone, our focus today is on the difference between sexual and asexual reproduction, and specifically how sexual reproduction produces genetic diversity inside of a population, whether it's a population of humans, dogs, plants, whatever it is. Sexual reproduction produces genetic diversity among those populations. And that's very important in order for the population to be able to survive in an ecosystem. So as you can see, the process that we're going to focus on today is meiosis. You haven't heard of meiosis just yet, but it's very similar to mitosis, which we've already covered. So everybody is different in some way, and those differences come from our genes. Now, everybody is unique. Everybody's genes are different from each other, and so that makes us ask the question, if we got our genes from our parents, and our parents have specific genes that they got from their parents, why is it that we're so different from each other even though we have the same basically set of genes as everybody else? There are small differences that make that difference between one person and another. Now, when we're talking about asexual reproduction, asexual, you have that prefix a, meaning without. That means asexual reproduction is reproduction where the organism does not have to have sex in order to reproduce. This happens in bacteria, this happens in some protists, and so these organisms will basically divide on their own. There are different types of asexual reproduction processes that we'll talk about later. Today, we're focusing more on the sexual reproduction, where there is sex involved between organisms in order to produce an offspring. So that introduces what we call gametes. Gametes are specialized cells in our body that are used for sexual reproduction. Now, all of the cells in our body are very specialized. We have muscle cells. Those muscle cells help us move around. We have digestive cells that break down food and transport it into our blood, or transfer it into our blood, rather. We have cells in our lungs that help us to take oxygen from the air, put it into our blood, take carbon dioxide out of the blood, release it into the atmosphere. And so all the cells in our body are very specialized. And when it comes to the sex cells, these sex cells are very specialized as well. And actually they are different from other cells in our body in many different ways. We call these sex cells gametes. Now, multicellular organisms will use gametes in order to produce an offspring. In humans, we have the sperm and the egg. Those are the gametes for humans. The egg, we call that the ovum. Now, all the other cells in our body, the ones that aren't used for sexual reproduction, we call those somatic cells. And like I said earlier, there is a difference between a gamete and a somatic cell. So let's look at those differences. On the left, we have somatic cells. Those are our body cells, the cells that make up the rest of our body. And the main characteristics that define those are: one, they're body cells. Two, that these cells are produced through the process of mitosis. The nucleus divides, and inside that nucleus, we have two sets of chromosomes. That's what diploid means. Whenever you see that word diploid, it's referring to the number of chromosomes, and in diploid organisms, they have two sets of chromosomes. That prefix di means two, so we have two sets of chromosomes. Those are diploid cells. And then through mitosis, as the cell divides, we're producing two daughter cells. All right, cytokinesis happens and the cell divides and we have two daughter cells that are produced. All right, so that's mitosis, that's somatic cells. Let's look at gametes. With gametes are sex cells. Again, the sperm or the ovum. These cells are produced through meiosis, the process of meiosis, which we're going to talk about here soon. It's very similar to mitosis. There are a lot of similarities, but there are key differences that make this process different and very important for the survival and to produce genetic diversity. Gametes are haploid cells. Diploid means that they have two sets of chromosomes. Haploid means that they only have one set of chromosomes. And we'll look at what that looks like here in a bit. I have some illustrations. I have some animations embedded here that will show you how we go from a diploid cell into a haploid gamete. And finally, through this process of meiosis, instead of producing two daughter cells through mitosis, here this process produces four daughter cells. So let's look at meiosis, the process of meiosis. This is another process where we have nuclear division. Remember, mitosis is not dividing the cell in half. Mitosis is not splitting the cell. That's cytokinesis. Meiosis is the same way. It's not splitting the cell. It's not producing the four different cells itself. It is producing four different nuclei in order for all of those daughter cells to have their own nucleus. Those nuclei produced through meiosis are haploid, and so they only have one set of chromosomes. Now, I said that the process is very similar to mitosis, and the way that it's similar is that it uses the same phases. Remember, we've gone through and I've given you a few different ways to remember the process of mitosis. And we have first to remember the phases, and the phases in order. We have the story. Remember, we have a pro athlete. That pro athlete met a girl. Her name was Anna, and she gave him her telephone number. So those steps are the steps of mitosis. Prophase, pro athlete. Metaphase, met a girl. Anaphase, her name was Anna. Telophase or telophase, she gave him her telephone number. I also gave you the hand signals to help remember what happens during each of those phases. Remember, we have prophase, where all the chromosomes are together, the nucleus dissolves. In metaphase, those chromosomes go to the equator of the cell. And then from there, anaphase, the chromosomes or chromatids, sister chromatids, are separated and pulled apart, going to opposite sides of the cell. And then telophase or telophase, we have the chromosomes on opposite ends. So again, prophase, metaphase, anaphase, telophase. We see those same steps, those same phases in meiosis, but with a few differences. Instead of going through each of those phases once, in meiosis, the cell goes through each of those phases twice. So there are kind of two cycles to meiosis. Instead of one cycle in mitosis, it's two cycles in meiosis. So here with meiosis, we have prophase 1, metaphase 1, anaphase 1, telophase 1. Then the cell splits. We have cytokinesis, and then we have prophase 2, metaphase 2, anaphase 2, and telophase 2. Now over here, I have a diagram that shows each cell in shows a cell in each of those phases. I don't expect you to be able to see those right now. If you're following along in the with the PowerPoint that I've posted, then you can see those different the different cells, the different phases, and you can see what's going on a little bit clearer. I'll have pictures along when I show and describe what happens during each phase, and so you'll get a lot more clear idea of what's going on. So let's go into it, and let's start off with prophase 1. Prophase 1 is extremely important when it comes to genetic diversity. In prophase 1, just like with mitosis, the nuclear envelope dissolves. We have the chromosomes that form, and here, a difference between meiosis and mitosis, we have what are called homologous chromosomes. Homologous chromosomes are chromosomes that are very similar in size, shape, and contain similar genetic information. So a gene that you have for maybe, let's say, eye color, you'll have 2 chromosomes that will contain a gene for that specific characteristic. All right, and so one of those genes you get from your mother, one gene you get from your father. And so during meiosis, prophase 1, those homologous chromosomes will pair up. They'll come in contact with each other and they'll pair up. And what they do is, it's an extremely important step in prophase 1, is they will actually exchange parts of their gene. This is called crossing over. So here we have 2 chromosomes, and these are homologous chromosomes. These chromosomes will kind of wrap around each other. They'll come in contact, and they exchange parts of the chromosome. They exchange segments of DNA. And so this first chromosome will now have a kind of red tail where it used to be blue, and then it gives that section of DNA over to the other chromosome, the homologous chromosome. And so now we already have an exchanging of genes. So a chromosome, genetic information that you got from your mother, has now mixed with genetic information that you've gotten from your father. So your mother's chromosome, your father's chromosome, they pair up, they come in contact, and they exchange information. That's called crossing over. After prophase 1 is metaphase 1. Just like in mitosis, during metaphase, the chromosomes line up on the equator. Now this time, instead of single chromosomes lining up on the equator, we have homologous chromosomes, we have homologous pairs that line up on that metaphase plate. During metaphase, we have spindle fibers that start to form, and they'll connect to the chromosomes, connect to the centromeres of those homologous pairs. During anaphase 1, we have spindle fibers that start to shorten. Remember, I gave the analogy that the centrioles on either side of the cell, those are like a fisherman. The chromosomes are fish, and then the spindle fibers are like the fishing line. And so as the fisherman casts its line, catches a fish, it starts to shorten the fishing line. Those spindle fibers, it pulls that fish closer. The spindle fibers shorten, and it pulls the chromosome to the side of the cell, a pole of the cell. And that's what happens during anaphase 1. But here, instead of those spindle fibers being connected, two spindle fibers to one chromosome and separating sister chromatids, here the spindle fiber is connected to one of the homologous pairs. And so the chromosomes, the homologous pairs, are what separate during metaphase one. I'm sorry, during anaphase one of meiosis. And so here we have a spindle fiber that's connected here, a spindle fiber that's connected to the centromere here. And so as those spindle fibers shorten, this chromosome is pulled to this side of the cell. This chromosome is pulled to that side of the cell. And so we have a separating of chromosomes, separating of homologous pairs. So we've already started to make the daughter cells genetically different from each other. During telophase 1 is just like mitosis. We have the chromosomes on either side of the cell, either pole of the cell, and now the cell starts to split. It's ready to divide into two daughter cells. So during telophase 1, we have the first time that cytokinesis begins. Remember, we're producing 4 daughter cells through meiosis, and so cytokinesis is going to happen twice because you start off with one cell, it divides once to produce 2 daughter cells, and then those daughter cells will go through meiosis, through those phases of mitosis again, and then those 2 daughter cells will split to produce 4. So that's how we get 4 daughter cells in meiosis. So let's look at an animation of the first part of meiosis, that first cycle, that first phase of meiosis. Important thing to note is that the chromosomes, as they are pulled to either side of the cell, those chromosomes still have 2 chromatids, sister chromatids that make up the chromosome, and that becomes important in the second phase, because if we didn't have that, we wouldn't be able to go all the way through the second phase or the second cycle of meiosis. All right, so let's look at that animation. And so here's our animation. We see here homologous chromosomes lined up along the equator. They were paired up, split during anaphase 1, and now we have 2 separate daughter cells. Each daughter cell has the chromosome, one chromosome that belonged to the set of homologous chromosomes. So here in this example, we have a long chromosome and a short chromosome, one in each cell. Now, during prophase 2, again, it's prophase, so we're starting off with the chromosomes bunched together. Here, the chromosomes will be formed. We have spindle fibers that start to elongate, and the nuclear envelope is dissolved. During metaphase 2, those chromosomes line up on the equator of the cell. This time, the spindle fiber connects to both sides of the centromere so that during anaphase, those sister chromatids can be pulled apart. So here, we have the chromosomes on the metaphase plate, and they're all lined up single file instead of paired up like they were during metaphase 1. During anaphase 2, those spindle fibers, again, we have the fisherman that starts to reel in the fish. That fishing line gets shorter, the spindle fibers get shorter, and that pulls the sister chromatids apart, and they start getting pulled to either side of the cell. And then telophase 2, we have one sister chromatid, now a chromosome, in each side of the cell, each pole of the cell. And so the nuclear envelope forms around those chromosomes, and we have a new nucleus. That nucleus has different genetic information in every single one of the daughter cells. In mitosis, the daughter cells that are produced are identical to each other, and they're identical to the parent cell. During meiosis, we have a mixing up of genes, and then we have a separation of common genes called alleles. We have a separation of alleles into 4 different daughter cells. So every single one of those daughter cells is genetically different. And this is why we are different from everybody else in the whole world. Even our parents. Even though we're getting our genes from our parents, we're different from them because through the process of meiosis, we have a mixing up of genes that causes us to be different. So here we have another animation. This is for the second cycle of prophase. We see sister chromatids are separated and cytokinesis happens again. Now we have 4 daughter cells. So as I talked about just a second ago, the process of meiosis is extremely important because if we didn't have the process of meiosis that mixed up the genes during crossing over is extremely important. Remember that. That initial exchanging of genes between homologous chromosomes, if we didn't have that, then we would all be the same. Our genes, with the exception of mutations, our genes would all be the same. We'd all look the same. We'd all act fairly similar. Nature does play a role in how our genes are expressed. We'll talk about that as we get into epigenetics. But every single one of us is different because the process of meiosis mixes up our genes during crossing over. And then during anaphase, a little bit during metaphase, because that's when the chromosomes line up, and there's a 50-50 chance that homologous chromosomes will be on one side versus the other. But prophase 1, that crossing over is extremely important. And then during anaphase, when chromosomes are being pulled apart and separated onto opposite poles of the cell, that's what creates the differentiation between the genes in one daughter cell versus genes in the other 3 daughter cells. So all of this is extremely important. And when we get into the ecology unit, we'll start talking about why genetic diversity in a population is so important. All right, populations wouldn't be able to grow and survive as well as they do if we had everything the same. And so genetic diversity is extremely important. This process of meiosis to help sexual reproduction is extremely important for survival of the populations. In your Google Classroom, you will see a follow-up task for this lecture. Go there and answer those questions on Flipgrid, and I look forward to seeing your responses. Thanks.